Hello students, Mrs. Kleins here with your first section of notes in our genetics unit. In this section, we're going to talk about Gregor Mendel, important terms and concepts, and monohybrid crosses. Let's first discuss the origin of genetics. As you remember, the discovery of DNA took some time, but once we understood its structure, we learned that DNA segments contain this information that we call genes, and genes code for the genetic traits of an individual. Remember that specific genes can code for certain proteins, which can lead to different expressions of those genes. So, when a gene has more than one variation, we're going to call that an allele. And you'll hear these terms used a lot, so I just want to really quickly tell you how they're different. So here is an example of an allele. The gene for eye color. Not every individual has the same allele for eye color, but there is a gene that codes for a protein that breaks down the webbing between fingers during the early stages of development, and you know both parents have that gene. So if it's a gene that's always present on a chromosome in the same form, there's not going to be any variation. For this, we're not going to really have to do any predictions based on whether or not the offspring will express that gene. But if there is a variation in a gene, that's when we call it an allele. This could include hair color, eye color, and other traits that are unique among us. So, genetics by definition is the study of patterns of heredity. This means studying how traits get passed from one generation to the next. So for heredity, we can observe patterns when we study parents and offspring or many generations over time. So where did this start? We give a lot of credit to this person here. You may remember him. This is Gregor Mendel. He lived from 1822 to 1884, so kind of ahead of his time, well before the understanding of the structure of DNA. He did discover in his studies and concluded that one form of a gene can mask another. He worked extensively with garden pea plants, and whenever he was cross-pollinating these different pea plants in the monastery that he worked, he noticed that certain traits could be controlled whenever he bred specific parents with specific traits together. So he started to notice some patterns of heredity based on the traits of the parents. He also learned that some genes could be masked by other genes, and that explained why sometimes we would see traits skip a generation. So looking here with flower color, he could breed a purple with a white flower, and the generation that's produced, the first generation, we call this the first filial generation, all had purple flowers. But whenever he cross-pollinated two of the flowers from the first generation, he noticed in those offspring the reemergence of white flowers. So he was noticing that genes can be masked or hidden in some individuals. He also developed, based on these observations, 3 laws. The first law that we'll talk about is the law of segregation. What that means is that during meiosis, each parent contributes one allele for a particular gene or genotype, and those genes during meiosis get separated from each parent. So remember, we get a copy of each gene from each parent, and during meiosis, those get shuffled up and segregated into different gametes here. So it kind of happens at random, but these get separated, meaning you get just one copy from each parent. The law of independent assortment can also be seen here, which is that alleles get sorted independently into gametes during meiosis. So, receiving one allele for one trait and receiving another allele for another trait that don't necessarily correlate with each other unless they're on the same chromosome. But if they're different genes on different chromosomes, those get shuffled around kind of at random during meiosis. So the allele a gamete receives from one gene will not necessarily influence the other genes. You can see here, in this case, I've got true breeding round and yellow seeds, and then true breeding wrinkled and green seeds. The seeds on the left will always contribute these alleles, either a big R or a big Y. The seeds on the right will always contribute the little r and the little y. But the individual under the gametes, they'll either contribute a big R or a little r, and then either a big Y or a little y. And how the R's get shuffled around will not affect how the Y's are shuffled. Okay, in this picture, you can see that we've got to account for all possibilities because this can all happen at random due to segregation and independent assortment. So these different alleles can assort independently into these gametes in all of these possible combinations. So when you're doing genetic crosses, you've got to consider what are all of the possible outcomes. Gregor's third law is the law of dominance. Dominance means that for each trait, there is one gene that can potentially mask the expression of the other. The dominant gene will always mask the recessive. So you see in this case with the tall plants, tall is the dominant trait and short is the recessive trait. So if the offspring gets at least one copy of the dominant gene or allele, they're all going to express the tall trait. You've heard these terms. Now let's get some information down about them. First is genes. A gene is the unit of heredity. Genes are found on chromosomes, which are made up of DNA, and genes are expressed as proteins. Genes can vary in size and complexity. So for example, looking at one gene for flower color is on one specific chromosome in that individual's nucleus. An allele is an alternate form of a gene, which means there's possible variations for that gene, whether it's flower color or seed shape. But anytime there's differences among the offspring of parents, or if parents are different from their offspring, we know that there are alleles at work. A few more important terms are dominant versus recessive. Dominant alleles are always represented in genetics as a capital letter. Usually, the letter kind of corresponds with something that you're measuring, so it's easier to track. Recessive alleles are going to be represented by lowercase letters anytime you write the genotype of an individual. Genotype is going to be the alleles it has. You'll always put a dominant allele in front if present. In this case, the individual has both dominant alleles. In the next case, the individual has one dominant and one recessive. So you will always write the dominant allele before the recessive because you know the dominant allele will take over the recessive trait. The only time the recessive trait is expressed is whenever there is no dominant allele present. So the individual has to have both copies of a recessive allele. When you see genotype versus phenotype, genotypes are the combinations of alleles that the individual can inherit. So we always express those in letters. The phenotype is the physical expression of the trait. So what do we see? So we will say the genotype is little b, homozygous recessive, and the phenotype would be a white flower. In this case, we would say the genotype is big B, and the phenotype would be a purple flower. On to homozygous versus heterozygous. The prefix homo means same. So when an individual has two copies of the same allele, we would say that they are homozygous for that trait. We also call these pure breeding alleles, which means that this individual will always contribute one specific allele when it is bred with another individual. If we say the individual is homozygous dominant, that means they've got two dominant alleles. If they're homozygous recessive, they've got two recessive alleles. Remember, with homozygous dominant, they will express the dominant trait. Homozygous recessive will express the recessive trait. Heterozygous individuals, we sometimes call those hybrid alleles, which means that they get one copy of each. The prefix hetero means different, so they have one dominant and one recessive allele. But as you know, the dominant trait will mask the recessive, so even though their genotype is different from the homozygous dominant, the traits that they express are the same. Punnett squares are mathematical tools developed by Reginald Punnett, and they're used to predict probabilities of outcomes involving parental crosses. So if you're looking at one simple trait like pea pod color, as seen on this slide, you can use a Punnett square to predict the possible outcomes or phenotypes of that offspring, and their possible genotypes as well. We can express these results in the form of ratios. Phenotypic ratios are going to be where you say, what do I see in the offspring? In the case of a phenotypic ratio for this specific cross, I would see 75% of my offspring have green pods and 25% have white. Some individuals will express it also as a ratio of the number of squares inside. So a 3-to-1 ratio, since there are 3 squares that have green and 1 that has white. Genotypic ratios are done similarly, but you're looking at the genotypes. So in this case, with this Punnett square, each box represents 25%. 25% of my offspring will have the homozygous dominant phenotype. 50% will be heterozygous, and 25% will be homozygous recessive. A monohybrid cross uses Punnett squares, but we are just looking at 2 organisms mating or crossing with different variations of alleles for one specific gene. So you use Punnett squares to complete monohybrid crosses. Here's another example showing you how to do that. I've got my parent alleles here on the outside. The letters on the top are from the father. The father was heterozygous. The mother was also heterozygous. And her genotype is written on the left side of the Punnett square. You can see when you cross them, you complete the inside of the Punnett square with the genotypes, pulling 1 letter from the top and 1 from the side. So in this box, I would pull my 2 alleles and get homozygous dominant. In this box, the individual will get a dominant allele and a recessive allele. Remember, you always put the dominant allele in front of the recessive allele. In this box, again, 1 dominant and 1 recessive. Then in this bottom right box, both of the alleles that merge will be recessive. So I would express my ratio similar to how you saw earlier, where I list how many boxes would give me each genotype and phenotype in the ratio, along with the percent chance of each genotype and phenotype. This is the end of this section of our video notes. Be sure to go back and watch this video again if needed, and don't be afraid to ask questions as we work through practice problems in class.